Thank you very much, Julia, for your kind introduction, and all of you for showing me in the last few days how much has happened here in Chicago in nanotechnology. And it's amazing to see the programs and all the strength that has been built in the recent years. So why should you care about the mechanical design of proteins? So let's step back and just think. Your most romantic moments in life, you allow magic to unfold. A sperm is sitting in the egg, and you see how the flagellum of the sperm is driving the head of the sperm. It gets in contact with an egg, it's wiggling around, and then it happens. The sperm penetrates into the egg. Even though we all know it's happening, otherwise we wouldn't be here, we do not really know how it's happening. A lot of bits and pieces are coming together, but it's an amazing surface science problem. And you can see just from this video, it's not equilibrium science that would explain how a sperm gets into an egg. But even this event is happening far away from equilibrium. So what do we know today? So here you see a membrane of the sperm and the membrane of the egg. And there are two proteins that need to be there in order for the sperm to penetrate into an egg. If you delete those two proteins, nothing is happening. They have wonderful names. Izumo is the protein on the sperm surface. Juno is the protein on the egg surface. And they need to form a complex. Otherwise, nothing will happen. But then there are also a number of other proteins, and exactly how this is happening at the structural level is unknown, because the sperm can only penetrate if these two membranes are finally going to fuse. But then, once the sperm is inside the egg, you all know that only one sperm should penetrate the egg, but not more than one. So somehow there must be a mechanism in place that prevents further sperms to penetrate. And biologists have shown that there's a firework that happens as soon as the sperm enters the egg. Zinc ions are released, and it's called the zinc spark. And the zinc spark leads to a number of events outside of the egg surface, but exactly again how this prevents polyspermy is unknown. We know that if the zinc spark doesn't happen, a next sperm and next sperm and next sperm can penetrate the egg. So, what's happening after that? Well, you know, cells start dividing, they start dividing. First they form a clump of cells, then they start sorting each other, a lumen is formed. But until that point, we are still a little spherical clump of cells. Then this clump elongates, and here you see how the membrane is bending to the inside, and that you form a lumen there, and that finally defines our mouth and our after. So how can we use modern computational tools to learn a little bit more about the events that are happening? So luckily, the two proteins necessary were identified in 2016. The structures of the two proteins is known. The structures of the complex is known. And, but those structures are equilibrium structures. And you know, if you start growing a crystal, you even dehydrate a protein and you squeeze it, arrange it in the crystal. So what happens even to the protein structure if you hydrate it in water? How is it changing? So this is what we looked at using computational tools. And what we found was that this molecule Izumo on the sperm surface attaches to Juno in a flat configuration. And it fluctuates, if not bound to Juno, between a flat and a bent configuration. The bent configuration called the boomerang state. But then if we, and the ion binding sites on Izumo are known, if we insert divalent or multivalent ions into those binding sites, we see that Izumo is switching from a straight to a bent configuration, preferentially. And then we simulated what happens if we try to dock the bent configuration to Juno, and saw that its affinity is greatly reduced. So let me show you those simulations. I left away the water, and you see here the complex with Izumo flatly bound to Juno. Then next you see we docked the boomerang state to Juno, and you very rapidly see that it partially detaches. So you switch from a strong binding to a weak binding event. And then if we insert zinc ions, you see how Juno is switching from the straight to a bent configuration. So what we are proposing is that as soon as the zinc spark is released, you preferentially push Izumo from a straight to a bent configuration, and thereby make it much more difficult for the next sperm to even adhere to the egg surface. Something you would not predict just based on equilibrium crystal structures of these proteins. So with this, I wanted to introduce this concept, think about dynamics, think about non-equilibrium structures. Because life is happening far away from equilibrium. Yet, what we know in biology and in medicine is mostly based on equilibrium structures. Also, most of our screening assays screen proteins against protein ligands in the equilibrium state. And you know, the two major tools are X-ray and NMR. But now think about forces being applied to proteins. What do these forces do? And we can't see that with NMR or X-ray. But we can see it in simulations. Here you see a steered molecular dynamics simulation of a hydrated protein. And you see that even though biochemistry has established so many structure-function relationships, if you now pull on a protein, you change the structure. So you should also change the function of proteins by mechanical forces. So how does this field develop? Initially, I think a lot of in the community, a lot of people thought that if you apply forces to biological materials, they probably behave like polymers. But proteins have secondary structure. And most of our polymers do not have secondary structure, unless you really design it carefully. So these first AFM experiments, where Hermann Gaub and his team pulled on proteins, on single proteins, were really astounding, because you could see nonlinear behavior if you pull on multi-domain proteins. The first domain popped open, a second one, a third one, and you could see, as you pulled on the protein, these force spikes appearing. So proteins have a very interesting mechanical signature. And in the meantime, we know that the mechanical signature of proteins depends so much on their structure, on the angle at which you pull on them, and the inherent design of the protein. At that time, nothing was known. People thought that as these measurements were done on titan that connects in a muscle, the muscle fibers from slipping apart if you excessively stretch the muscle, that proteins in nature are used as shock absorbers, the key mechanical function. And so at that time, we started a collaboration with Klaus Schulten, who worked not very far from here, to ask, are there ways by which we can model the mechanical unfolding of proteins? At that time, despite him having the best computational resources of the country and probably the world, we could not even afford doing the simulation in a box filled with water molecules. Instead, we used little droplets. And we first pulled apart the same titan domains that were pulled in AFM. And initially, the thought was, oh yeah, mechanical stability and thermodynamical stability is equal. So thermodynamical stability of a protein is given by the number of hydrophobic residues that you bury inside the protein. But even this very first set of simulations showed already that what you need to break are clusters of backbone hydrogen bonds. And therefore, the angle at which you pull plays a major role. Many studies were then done to show that thermodynamic stability and mechanical stability are not the same. And you can imagine that immediately. Let's assume you form a cluster of hydrogen bonds, and once open the cluster this way, that should be a weak bond, or you shear them, and then all the bonds need to be broken at the same time. That's a strong bond. But we are not utilizing that in a single application so far. We make so many clusters of hydrogen bonds, but we are not thinking about how we can exploit that for engineering applications. So again, Hermann Gaub's group first looked into that and made these oligonucleotides, showing indeed that depending on the geometry in which you pull, you can peel open a cluster of hydrogen bonds easily, or the bonds would be much stronger if aligned. So we were wondering whether we can take advantage of that for a proof of concept study in engineered environments. And for that, and we were working for a long time on this question, whether we can build devices by harnessing the forces generated by biological motors. And the typical way of doing this type of work is that we take a surface, we put the motor protein kinesin on the surface, and then use the filaments. They fall onto the surfaces, and the kinesin movement is pushing the filaments forward. So we were wondering whether we could do the following experiment. We are all involved in fundraising. I heard so much about that the last 24 hours. So we were wondering, and since we moved to Switzerland, whether we could deposit gold nanoparticles at the Swiss border, and then use motor proteins to carry the gold nanoparticles from the Swiss border to ETH. So how do we do that? First of all, the gold nanoparticles need to be deposited in a stable manner at the Swiss border. They shouldn't swim away. So we need to create a thermodynamically strong bond, sufficiently strong to withstand thermal motion. But then the bond needs to break as soon as a microtubule is hitting the particle. So you see, therefore, the two different geometries. The zipper geometry, by which we deposited gold on the Swiss border, and then the shear geometry, by which we then, as a microtubule picked up a particle, deposited at ETH. So briefly, what chemistry is involved, what forces are typically involved. And the challenge is really, you have a thermodynamically stable bond, yet in the moment of the collision between our molecular shuttle system and the gold particle, you need to pick it up. It needs to happen very rapidly in order then to transfer it to our unloading station. Let me show that this is happening. Here you see the time in different colors of a molecular shuttle coming in, picking up a nanoparticle, transporting it across space to ETH. And that this is really happening, you see it here. So you can indeed, just by utilizing the zipper versus shear geometry, you can create a mechanically active device employing the differences between thermodynamical and mechanical stability. So let's go back to proteins. We soon started asking the question whether proteins are really only utilized by nature as shock absorbers, or whether distortions of the structure can bring about functional changes. And here you see the fibronectin type 310 domain, the RGD loop. Jeff worked on the RGD loop a lot. In the 90s, he showed for the first time that if you take this loop out of a huge protein and put it on a surface at a certain density, that it allows cells to adhere and spread to those surfaces. So what we saw in these simulations was that if you start pulling on the protein, you straighten out the RGD loop. And we know the affinity of integrins for this loop goes down, whether it's presented in the loop or straightened out. But even more, cells recognize on our PET molecule, and I come back to that, whether this RGD loop is close to another site, the synergy site. And in order for the loop to bind into integrins, the synergy site latches on to the site of integrins. They have to be apart by 33 angstroms. If you now stretch these two domains, you increase the distance of the two. And again, this is now a bivalent binding motif. Mechanical force destroys that. So Jeff, again, incorporated these domains in hydrogens. So where to go from there? So these were the first structural predictions that forces can change the function of proteins. How can we prove that experimentally? At that time, it was very difficult to do. We got a number of questions. Typically, biochemists said, we got trained on printed material looking at equilibrium structures. Nature does not work under denaturing conditions, because if you stretch a protein, you partially denature it. Proteins might serve as shock absorbers, yes, but this famous but that you never like to hear when reports come back. And one reason this was asked was that the unfolding of biomolecules by AFM and optical tweezers at that time was done really rapidly. So high forces in the 100 piconewton regime were recorded, and people doubted that cells can therefore unfold a protein under natural conditions. So how to prove that for our pet molecule fibronectin? So we knew at that time that we can photolabel fibronectin using the cysteines, and then we attached a lot of fluorophores, donors, and acceptors, so that the energy transfer between donors and acceptors is reduced if you increase the distance to each other. It's called fluorescence resonance energy transfer. And if we added this labeled fibronectin back to cell culture, we could indeed see that fibronectin existed in many different conformations, as shown here in different colors here in the background. So with a series of papers, we could indeed demonstrate that forces generated by cells are sufficient to not only stretch but partially unfold proteins, or here, protein fibrils. Since then, we know that many structural motifs exist in nature that could be utilized, on the one side, to understand mechanoregulated processes in nature, but also many of you are chemists. There are a lot of design features where we need to think how to bring them to bear on synthetic molecules to make mechanochemical, mechanoelectrical materials with new functions. So I mentioned already this shear versus zipper geometry and how it can be exploited. We discovered using bacteria that catch bonds exist, which we published in 2002. People always thought up to that time that if a ligand is bound in a binding pocket and you apply force, that you accelerate the detachment. That is true if you think just of a key lock mechanism. But imagine force distorts the complex before the ligand can slip out. Then you can enhance the lifetime of this complex by pulling on it mechanically. I introduced briefly already that proteins might act as mechanochemical switches, and many more designs have emerged in recent years. So how do we now utilize and apply those principles to better understand, on the one side, biology, but also for technical applications in nanotechnology? So this is a slide very much to my heart, because it all started with proteins in equilibrium. I think it's well established today that proteins in various settings, wherever they are part of force-bearing networks, can be used and are used by nature as mechanochemical switches. And a lot of this research was done by the new nanotools and assays that have emerged in the last 20 years, and to which the community got access to by nanotech centers or institutes, as here. So nanotechnology and its tools enabled the emergence of a new field, which is called mechanobiology. And so with these nanotech tools, a lot of basic science discoveries were made now in biology. And the goal now is to utilize this wave of new insights for new medicine, new medical products. And we can call it nano or nano-mechanomedicine. And I would like to show you now where we stand with this approach. So we all know forces matter. We have developed a lot of nanotech scale assays, cellular assays where cells are sitting on flat surfaces, because there we could easily engineer the Young's modulus or spring constants or change the topography and so on. But somehow we do not yet know how to translate this to 3D. So people very often in the community just project this should happen, but without having the proof that this is happening. So why is it so difficult to bring this knowledge towards the clinic? One big problem is that we have no force or tension sensors that can be incorporated easily into tissue. So in a tissue environment, we do not know how much force the cell applies to its environment, how proteins are stretched. We have no way of measuring that so far. So we were wondering whether we can create nanotension sensors that are highly specific, that are biocompatible, and that are stable in blood. Otherwise, we don't even have to start. How to do that? Well, we worked a lot with bacteria and decided to take advantage of the co-evolution of bacteria with their hosts that has taken place now for more than 500 million years. Bacteria optimizing their chance to invade a host and a host developing mechanisms to prevent that. So we were wondering whether we can use bacterial adhesins, molecules, glue molecules, by which bacteria invade wound sites, because then everything is given. It's stable in blood, otherwise bacteria wouldn't have evolved these peptides. And can bacteria distinguish whether or not they face healthy tissue, or whether, for example, mechanically or enzymatically, whether they see a wound site where tissue fibers are cleaved? And what we want to bridge with a new sensor is our knowledge of single molecule mechanics, the fact that forces can switch the structure of proteins, and we would like to learn how this impacts embryogenesis, tissue homeostasis, regenerative processes, but also how it's changing in cancer, in fibrotic disorders, how we can exploit it for tissue engineering applications, and many more. So I mentioned our PET molecule already. It has many, many domains all linked together, and then it aligns and forms fibrils. And here at the N-terminus, there's a regime where bacterial adhesins bind to fibronectin. So wound sites are full with this molecule. Luckily, an NMR structure was available of bacterial adhesins binding to the N-terminal domains of fibronectin. And what you see very nicely is under equilibrium, this bacterial peptide binds to two domains. But then if you stretch fibronectin, you increase the distance between the two domains, and the bacterial peptide pops off from one domain. So it's like building with Lego blocks, an analogy that was used several times already in the last 24 hours. If you would stretch the base plate, only a fewer numbers of knobs can bind. So by tension, you switch the adhesion of the bacterial adhesion from high affinity to low affinity. Certainly beautiful to see that in simulations, but can we prove that experimentally? So I mentioned already our FRET probe, and then we labeled our bacterial adhesion with a third color. We added both of them to the cell medium. And what you see very nicely is the FRET signature of the extracellular matrix fibers that fibroblasts make in 2D. And then we see high FRET. These are regions where cells have not pulled so much on the fibers in red. And it's in these red red regions where we see that the bacterial peptide is binding. And then from this and many other images, we do a pixel by pixel comparison and see that two completely different readout mechanisms correlate nicely over a range of strains. So with that, we now have a probe available to maybe go into tissues. The important part to develop this probe is using FRET at the tissue level is essentially impossible because you have so much background signal from tissues that you can't interpret or quantify FRET ratios in meaningful ways. And where do we apply it to? I think probably the best studied disease now, where it has been shown that not only biochemical signals matter, but that the physics of the environment, be it now a Young's modulus, has a huge impact on cancer malignancy and progression. And what we know, though, is that many different factors can cause a tissue to stiffen up once a tumor is developing. And that includes extracellular matrix fiber cross-linking, fiber bundling in these tissues, enhanced extracellular matrix production, which is observed, and then certainly the solid stress that builds up by dividing cells in these densely packed tissues. So where and what can we learn from our probe? Well, we first sliced up a lot of healthy organs. We stained them with polyclonal antibodies of fibronectin. You see it in green. So a lot of fibronectin is present, but these fibers do not stain or bind the peptide, meaning that these fibers are under high tension in most of the healthy organs that we studied so far. But then if you start slicing up tumor tissues, and this was the first one we published, you see large areas where the peptide is binding, showing that in tumor tissue, and we did not expect that, we find large areas of structurally relaxed fibers. They could be cleaved or relaxed for other reasons, for example, simply by the upregulation of matrix production. We then started to correlate. Are these regions of relaxed fibers found next to blood vessels? No, we did not find that. And that was the next huge surprise that was not predicted. We find the relaxed fibronectin fibers close to alpha smooth muscle actin positive cells. And those are myofibroblasts. Myofibroblasts are more contractile than the fibroblasts that you find in healthy tissues. Myofibroblasts are there to contract the wound site and close it. And so they produce a lot of matrix, create a lot of force, and we thought that in their proximity, fibronectin fibers should be stretched, not relaxed. But something else we find in the proximity of these structurally relaxed fibers is thick bundles of collagen. Usually in healthy tissues, you find collagen, but not in thick bundles. And we find the relaxed fibronectin fibers next to collagen fibers highly stretched and highly bundled and straight. So we think that the cells are switching from pulling on fibronectin to pulling on collagen fibers in these regions. So is this of importance? We, in recent years, now studied many proteins that are known to bind to fibronectin and asked the question whether or not their binding to the fibers is up or down regulated as we stretch fibers. And therefore, we focused on one cytokine. It's called IL-7. And this cytokine is the only one that does not activate the immune system. It promotes B cell and T cell proliferation and maturation. If you lack IL-7, This causes immature immune cell arrest. The IL-7 concentrations are inherently low, yet all the cells compete for IL-7. And so we were asking, does IL-7 bind better to stretched or relaxed fibers? And what we found is that IL-7 binds better to stretched fibers than to relaxed fibers. This is really interesting because we also looked at lymph nodes. And we found in naive lymph nodes and adjuvant swollen lymph nodes that fibronectin fibers are under huge tension. But then if you have virally infected lymph nodes, you see that fibronectin fibers are cleaved. So taking these two together, we would like to propose that viral infections promote the release of IL-7. And indeed, if you looked at COVID patients, you see an upregulated circulation of IL-7 in their blood. And people so far think it's upregulated, its expression is upregulated. We think it's released from lymphatic tissues. Then we looked into matrix tracks that were observed in many mouse tumor models. And what we, through characterization of many compounds, found was that these matrix tracks are surrounded by basement membrane, which people had not stained for, and that this basement membrane has a vascular origin and not a duct origin. These tumors are rapidly grown in mice. We further found that, and are postulating that these vessels undergo an endothelial to mesenchymal transition. And these channels are filled with CD8 plus T cells, with M2 macrophages, that's not the pro-inflammatory macrophage, with relaxed fibronectin fibers, with collagen bundles, and tenascent C. So again, you find the presence of relaxed, and we are wondering what the function is. So since a lot has been learned on mouse that could not be applied to humans, we are also now investigating human breast cancer tissues. And again, we find that in invasive tumors that a lot of more relaxed fibronectin fibers are being found. So to establish a causal relationship in 3D, we are growing microtissues. And here we use a very simple platform of making scaffolds, seeding them with cells, and then we allow in between microtissues to grow. What we found is that in the growth front here, in the front of these tissues, this is where cells divide. And they are enriched with myofibroblasts. And then very rapidly behind this growth front, you see that alpha-smooth muscle actin, the signature of myofibroblasts, is disappearing. So they convert to fibroblasts. The growth front is only 10 to 20 microns thick. And this transition from myofibroblasts to fibroblasts happens within a micron or two. So we looked very much in detail how this is happening, what proteins are found as function of distance from the growth front, and put together a really interesting picture. It's not one factor that switches myofibroblasts to fibroblasts. It's not TGF-beta. We see this happening in the presence and absence of TGF-beta. Not so in 2D cell culture. So what is required to switch myofibroblasts to fibroblasts? It's not one protein. It's an amazing crosstalk between the cells, the matrix they make, and going back and forth and back and forth. And I don't have the time to explain this in detail. But what is important is that if you block any of the molecular players that play a role here, the maturation of tissue is hampered, and we see even deep in the tissue myofibroblasts. And the appearance of myofibroblasts is always a bad sign for when it comes to tissue repair and remodeling or fibrotic diseases. So what we need to understand is how proteins act as mechanochemical nanoswitches. This nanoswitch idea played a huge role in us understanding what's happening at the growth front. We think that forces play a huge role in the synchronization of cells in cell niches in many different organs. And we have to understand how this is all playing together. And what tips the balance, whether a tissue heals or undergoes pathological transformations. In order to get to the clinic, though, what is important is, can we inject this peptide into a mouse? Would it be stable? And would it accumulate in diseased organs? So here we radiolabeled the peptide, injected it into the mouse, and you see how it accumulates and stays for days to see in these two diseased ECM niches due to tumor growth. So learning how to best target the extracellular matrix too, not just the cell, is totally important. It is, and sometimes I feel in the cancer community, hearing talks, they don't even mention the existence of matrix. It's like describing the behavior of a spider by ignoring the spider net in which the spider resides. So all our cancer therapies are meant to kill the cancer cells. But even that leaves diseased matrix behind, and we are not thinking about the implications of that. So with this, we hope to open up new ways of thinking of tumor, cancer progression, fibrotic diseases, and have a tool now in hand to target diseased matrix. All antibodies target a protein, but we can target a protein and its physical state. So with that, I would like to thank you for your attention, many group members over many generations who contributed to the work that I could present today, and our clinical collaborators. Many thanks.